Because after the small coffee break, we are now resuming the session. I would like to welcome again all participants, mostly because maybe new ones are coming, but also to the ones who are following us live in webcast, in the web. Also, I would like to explain again the procedures. We will have 2 presentations, 2 minutes of technical questions of clarifications, and remember that we are looking forward to keep at least those 20 minutes for panel discussions. So your questions in more general format should be preserved and reserved for that panel discussion. I would also like to say that I'm a chair. I would like that whoever wants to make questions for my speaker, which I will introduce shortly, I would like you to really raise your hands and make sure that I can see you and see how many questions I am going to expect for him. So I will organize then better my timing so everybody has a chance to make those questions. Now after the coffee break session, it goes to more practical examples. We have a quite good framing of the problems that we are facing today, but now we are going to have 2 very specific examples of how, in practical ways, decisions needed to be made at the end. So the first speaker is Dr. Nigel Walker. He's actually British-born, but United States citizen, so he's really transatlantic cooperation, global kind of person. He's Deputy Director for Science for the Division of the National Toxicology Program, which is a research program at the National Institute for Environmental Health Science at the NIH. The program is very interesting, and I think it would be a very good example to show how things can be done in a kind of learning good practices, learning from examples of others that have experience prior to this. Because this program is a good example of intergovernmental cooperation. It's interagency government, but it's a research program that allows cooperation and coordination, particularly for toxicology testing programs within the federal government, but also has a cooperation between academic scientists and regulatory scientists. And also they want to forward-looking to see what are the challenges in the future, and they are already trying to integrate assessment through multiple databases and through different systems. So I think we are really looking forward to listen to you, Nigel. Welcome, and the floor is yours.
Thank you very much, and I really appreciate the invitation to come and discuss with you all about the National Toxicology Program. As you can tell, I am British-born, even though it's the USA, and they were really messing with your heads today with all the accents. So let's get this started. So the title that the organizers gave was, as you can see, a very operational title, The Lessons Learned from the NTP, which is where I've been for the last 20 years. And so I'm going to walk you through several things. I'm going to give you a very detailed overview of the NTP. I will then give you some case examples that I think highlight some of the issues that Bernard highlighted earlier on about the translation of science into policy and how we can make that more effective. And then I'm going to tell you about some of the new approaches that we're engaged in as science is progressing to actually do things a little faster, a little more responsively. And then I'll give you some final reflections on some of the lessons learned from my time there. So the NTP is somewhat of a unique organization in that it is an interagency coordination between several parts of the US government. We are based at, headquartered at the NIH, which is a biomedical research organization, NIHS, but it also encompasses other parts of the federal government, notably the FDA and the National Center for Toxicological Research, as also the Centers for Disease Control and NIOSH, who deal primarily with occupational exposures. So between FDA and NIH and CDC, we cover occupational, environmental, food, drugs. We can basically cover all parts of the regulatory spectrum. And our mission as an organization is really to evaluate the agents of public health concern by developing and applying tools of modern toxicology and molecular biology. It's a very contemporaneous mission because as science moves on, you can use new science quickly to actually make your decisions. So even though I'm at a safety assessment, a risk assessment talk, I always like to point out the fact that we actually don't do risk assessments. We generate hazards. As you can see in the upper left quadrant, a highly hazardous situation where the magnitude of hazard meets a very high exposure. And so one of our roles within the NTP is to really identify hazards and to characterize magnitudes of hazard. We don't do all the integration stuff for the risk assessment. We leave that to the policymakers. And how do we actually do what we do? We have a variety of different approaches. We have what we traditionally call the hazard characterization program. This is used in looking for carcinogenicity, toxicity at various life stages, primarily via in vivo GLP guideline studies that can stand up to scrutiny and regulatory vigor. We also develop new approaches, such as the Tox21 initiative and high-throughput screening, as well as other non-mammalian systems like C. elegans and zebrafish. And also we have a very strong emphasis on developing alternative models and the incorporation of non-animal models through our actions of NICETM and interagency coordination. We also do pathological research, this is what we call the research arm of the NTP. In addition, we also do literature-based assessments, which are somewhat similar to what EFSA does in terms of bringing together data from multiple data streams, be it for the identification of carcinogens, which is our congressionally mandated report on carcinogens, or for non-cancer health effects, which come now through the Office of Health and Assessment and Translation through a new systematic review methodology. And every once in a while, we do some exposure assessments, but those are really to contextualize the potential compounds that we're going to look at. In terms of a food safety areas of emphasis, we cover the whole gamut, from botanicals and dietary supplements to contaminants that get into the food chain, to actual mixtures, combined exposures, specific compounds such as PAHs and PACs. It really runs the gamut of different exposures, so we aren't focused on any one particular sector. So operationally, I like to think of what we do in 4 phases. We get problems that come to us externally, and we also identify them internally. We call these nominations, and that's very important because it really frames the area and the issue or the chemical that we need to look at. We then develop our solutions, which we do internally, but those strategies and the concepts of what we're going to look at, we then communicate externally to our stakeholders, to the public, to our regulatory partners, to make sure they're going to be responsive to what the original problem was. Then we get into the needs, which is very much scientific in that we design our studies, the different kinds of assays, different kind of endpoints. And then finally, how do we actually do that is very different than a normal academic or research laboratory in that we have contracts, interagency agreements that cover the span of different types of capabilities. So we actually contract out pretty much all the work that we do to make sure that we are not bound by our in-house capabilities to actually address a problem. In terms of how we deal with prioritizing the questions that come to us, we have quite a formal nomination process where compounds will come in or issues will come in from federal partners, from the public. Anyone in even you in the audience could go to the NTP website and nominate and say, we should be studying this chemical for this reason. And we would take that into consideration, we would look at it, we would look at the data that's available and see what the problems are and if we need to address anything. And so we get nominations both not only from the US, but also from other parts of the world as well. They then go through a rigorous process where we identify, we work with our agency points of contact externally to make sure that we are coordinating, and that all then gets put out into the public domain. We request solicit comments particularly on our literature-based health assessments, but also on our research strategies for looking at a given compound. We take all that advice on board as we then develop the different research programs, be it a research program or a literature-based health assessment. So this is all very transparent. We try and be as open as we can without allowing it to be unduly influenced. And essentially, this allows us to make sure that we're being responsive to the questions that are coming into us externally. We're primarily externally driven, not internally driven. And as you can see, I'm just using this as an example over a 10-year period from the 90s through the 2000s, that we had about 180 single nominations, But more recently, we've had much more what we call complex nominations. These may be complex herbals, combined food exposures, issues that cover multiple classes of compounds for which there wasn't necessarily a regulatory mandate to provide that safety data when it was originally submitted. The sources, as you can see, internally at NIHS, we only generate maybe 21% of our nominations through looking at research needs documents or our own ideas from the literature. Primarily, all the others are from our other NIH partners or from other regulatory agencies such as FDA and NIOSH and the US EPA and other folks such as Consumer Product Safety Commission and OSHA. Next. So how do we actually prioritize what we do? Because as you can see from the cartoon in the lower right corner, everyone's issue is their own priority. And so trying to balance the different priorities that people have for their given compound or their issue is actually very challenging. Often we need to be responsive. We have to, in some cases, make sure that we're going to be doing things by GLP because we want the data to be very robust. In other situations, we may not actually have the capability to actually do the research that we need to do. There may be fiscal constraints, there may be time frame constraints. And how creative can we be? We've heard about the difference between pure and impure science. My hands up, I'm an impure scientist. I try and do stuff that is going to be usable by a regulatory agency. So most of the time, my scientific creativity is out the window because I have to make sure that I generate data that is going to be usable by somebody. And so, but that's an interesting consideration when we're looking at a prioritization. The other thing that is very different about the NTP is that we are very much a team science model. And this is not collaborations. I want to dispel that straight away. This is actually ingrained within the NTP organization, and we're maybe 100 people, 50 PhDs, is that while we have an organizational structure, all the projects and everything we do transcends that structure. So teams come together in a very matrixed way, and you just bring the capabilities and the expertise you need to formulate the problem as needed. It's not a collaboration, it is the way we work. So very interested in that as an individual, you aren't given your own money to do a single project in isolation in your own research group. It's very much a team mentality, team science model. So like I said, it's very project-centric. We're all about the project, not the individual scientist. We don't get funded individually as PIs. We have a central funding that is the whole of the organization. And then we have to build consensus of the way we design a project to make sure it's meeting the need of the original nomination and is going to be useful. This goes, like I say, goes through consensus decision-making, which sometimes can be very arduous, because you have to come to agreement that this is the right thing to do. And you can probably tell, you put 10 scientists in a room figuring out what's the right thing to do, and you'll get sometimes 10 different answers. And so this is very much a very ingrained process of how to actually come up with the right questions. And like I said, the research teams span the administrative structure. And to put this in a pictorial manner, we have project leaders. They work through what we call middlemen, called the contract officers' reps, who actually interpret the designs of the studies that we need to do to then actually go out to the different contracts to make sure they can make them happen. We don't fund a single contractor to do a single study for a single question. We bring together the different kind of capabilities that we need for the question, wherever it may be. And if we need to go through subcontracts, we do it that way. In some cases, this is through interagency agreements. We have a very long-standing agreement with NCTR for some very big animal studies where we need some of that creativity and interactions and close working relationships with FDA product centers. But in other cases, we have primarily commercial contracts with folks that often serve the pharmaceutical industry. And so this, at the end of the day, is our logic model. We have our inputs. We come up with our nominations, our designs, and hopefully then that gets to an increased awareness of the actual science that should be used. And then ultimately that leads going through external factors to actual citations in regulatory documents, be it through direct incorporation or through influence or through communication to the original stakeholder. And as you can see, it's quite a long logic model, and so that can actually can take quite a lot of time. So what I'm going to do now is switch, given the operational model, I'm going to give you several examples that highlight how we do what we do. And the first is going to be chromium-6. Now, Chrom-6, this is not a food issue, really, because actually Chrom-6 is a drinking water contaminant, but Chrom-3 is actually an essential nutrient, so it's somewhat food-related. This, back in the early 2000s, had a lot of media attention. Julie Roberts was in a movie called Erin Brockovich, some of you may remember. This was around the leukemia clusters in California that were believed to be related to contamination of the drinking water from chromium-6. They came to us, they had questions about the carcinogenicity of Chrom-6 versus Chrom-3, which is a non-carcinogen, doesn't do anything in terms of cancer. And we know that it's carcinogenic by inhalation, but this was drinking water, so they had a very specific question. And there was a total lack of adequate data to actually address that for them. The outcome of that program was that we found that it was carcinogenic, both sexes, rats and mice. We then subsequently did a health assessment evaluation of the carcinogenicity along with other data, and this was identified essentially and classified as a known human carcinogen by our report on carcinogens. And the impact of that was actually this data and the classification was actually used by California to actually create its public health drinking goal for chromium in drinking water. It was also used by the New Jersey State Department, because even though California was our nominator, because all of our data is public and we do everything in the public, New Jersey could take all that, they could go to the raw data and use it for their own decision making. So it allows, by virtue of being transparent with our data and our decision making, others can use it for their own needs as they need to. The downside of this, though, is that this is an example of why we need new approaches, because this was one of our fast-tracked compounds. It was one that we are very proud in the fact that the study ultimately led to a regulation. But the time from that original study to actually the impact on regulation was about 10 years. And I think, as one of the speakers said earlier, we're living a much more time-constrained world now, as we've moved into the information age. And so taking 10 years to generate information that can be used and then actually implemented just doesn't seem as satisfactory. And this is something that we have recognized, that the length of time it takes to generate high-quality apical data is very long relative to often some of the needs, which are much more short-term, 1 to 2 years. The other example I'm going to give of work we're doing is on bisphenol A. And I'm sure there's nobody in the audience that knows anything about bisphenol A. And as you know, this has achieved a high level of scrutiny, particularly in the US with the exposures coming from migration from plastics. The GLP studies out there showed pretty much only high-dose effects, but they were resoundingly criticized by many academics because academic investigations are showing at much lower levels, way below the regulatory levels, effects occurring in different analytical models, in different investigative models. And so we clearly now have two different sets of data. High-quality GLP data at high doses, investigative academic data at much lower doses. How do you actually reconcile that? Because people have tried, and it just causes more angst between all parties involved. So what we actually have created with our colleagues down at FDA and NCTR is what we call the Consortium Linking Academic and Regulatory Insights on the Toxicity of BPA. It's quite a mouthful, so go with the acronym CLARITY, because we're trying to create clarity for BPA. And really, the goal here is really to bridge this divide between GLP, high-quality guideline science, and academic investigations. And so we eventually created a consortium which is actually somewhat unprecedented within the federal government, to actually have academics working on samples and working closely with the federal regulators to actually address a problem. And the way this is essentially conducted is we have a core study which is conducted in the GLP with full evaluation of levels and a high level of rigor, but the samples then actually go to the academic investigators to actually do investigative studies, ranging from a wide range of molecular as well as anatomical and structural and functional kind of studies. But the cool thing about this is everything is blinded. So all the academic investigators know they've got samples, but they don't know what the samples, where they're from. They don't know whether they were exposed, whether they were unexposed. And so that level of blinding creates trust. Because then now, once the data goes into a database, because we have a public database for this, they can't get the data until it's been fully uploaded and then released. And so the regulators can actually go to the raw data if they actually wish to make a different analysis of the original information. And this is just probably too much detail, but it's an overview of the study. We're still in the throes of the project. It's actually the 2-year portion is finished. We're starting now get academic investigators are starting to publish some of their data. And really, what has been interesting about this project has been it's really shown the cultural differences between academic science and regulatory science. And it's probably the pure science versus impure science. Exactly the things that were said earlier on were exactly what we have seen with this. The lack of creativity, being able to be totally independent and just follow one's nose as an academic, the pure scientist, as opposed to being able to generate data that's very useful. And we're gradually trying to bring those people together, and it's kind of working. The involvement in the study design, belt trust, the sample blinding, as I pointed out, has been huge in starting to break down some of these barriers. And it creates constant communication. We could never do this if we simply put money out there and said, can you generate useful data for risk assessment on BPA in the normal, traditional sense of how you might do it through academic funding? It could never happen that way. In fact, we know that because we've done that, and that's what actually created the angst. And so hopefully this communication will lead us to bridging those two very different worlds. So the final examples I'm going to give you now are the new approaches that we're using, because I've given you these two which are very much founded in apical endpoints for risk assessment and decision making. But one of the challenges here is if you're at the left hand of this spectrum, which we generally, as NTP, had lived, we generated 500 cancer bioassays. Very useful for risk assessment because you've got somewhat limited uncertainty. You've got to translate between species. That's okay, I can handle that. Some dose issues. But the further you go down, away from integrated biological organisms down to molecular and cellular events, it's much harder to translate that to a human health hazard because you've got so much more complexity that you have to deal with. The flip side of that is that when you're doing apical assays, you're in the tens, maybe 100 kind of compounds a year. But some of the issues that we deal with now are mixtures, where I can generate 1,000 mixtures in a day simply by reformulating something. The issue of pesticides that have different formulations. I can't just go after one at a time because I'll break the bank. I don't have the time. So how do we move down the biological level of organization to increase our throughput, but at the same time provide the data that's going to be useful? And actually, the whole, this sets us up with this dichotomy because we generally only generate the data when there's exposure of concern. And often people don't generate the exposure of concern until there's tox data to tell you there's a hazard. And so we often get wrapped up in this cycle of, well, show me it's a hazard and I'll go and generate the exposure data. And so what we actually need to do is move in different directions to actually generate earlier information on whether where there may be exposure or where there may be hazard. And one of the key things here is if you're in the upper right-hand quadrant, it's a no-brainer you should study it. Lots of exposure, lots of suspicion of hazard. But what do we do with those that are in the upper left quadrant? We've got somewhat of a suspicion, but there's low exposure probability. Should we not study that? Maybe. What if you're in the bottom right-hand corner where there's lots of exposure, but we don't really have any good data to generate a suspicion of hazard? Should we just ignore it? We can't really do that. So that's why we've been generating data to move those into to a prioritization scheme. And one of the things that we did was about 10 years ago, we came up with a new vision for how we're going to do toxicology. This was initiated under Chris Portier, the director of the NTP at the time, and it's really to move toxicology primarily based in an apical disease-specific models to more of a predictive science. Now, the good thing is we said that this was the 21st century, so we've got like 100 years to do this. But the reality is that we are already moving quite quickly along this road. Part of that has been what we call the Tox21 Consortium. This is an interaction between FDA, NIH, and EPA on high-throughput screening. And the whole idea of this is actually to provide some of that suspicion of toxicity much more quickly. The initial phase, the proof of principle phase, was, can we even do this? Which we did. And then the second phase was to expand the screening to 10,000 chemicals in 100 cell line bioassay type assays, primarily based on nuclear receptor activation and stress responses. And now we're starting to incorporate metabolism and other types of assays. The whole point being here is we're rapidly moving to a point where we can generate lots of information on tens of thousands of chemicals to provide us that suspicion of toxicity. And the other benefit from this is we now can do read-across. Well, this is the potential to do read-across, where when you integrate this, you identify the fact that there are groups of compounds that, based on their bioactivity in vitro and in these shorter-term models, cluster together. And so this allows us to bridge from apical assays that may have been done on a legacy compound to new compounds or mixtures that we have no information on. And that really helps us prioritize. And we're using this currently particularly for dietary supplements and other mixture situations. The other example that I'd like to give where we've actually put a lot of this thinking into practice was about a year ago where, I don't know whether this made the news over in Europe, but there was a chemical spill in West Virginia where there was residents identified that there was a strange smell in the water. There was relatively quickly, there was an advisory. Turns out that 10,000 gallons of an oil dispersant was actually spilled into the river, and they hadn't really told anyone right away, and before you know it, it's everywhere. It's in the water, it's coming through the taps, and that's why they were smelling it. And so CDC relatively quickly issued a 1 ppm screening level, but it was really based on somewhat limited information on one or two chemicals. Didn't really address the mixture issue, didn't address some of the other compounds. And so they came to us asking us for a very specific question. Can you provide some data to actually address this uncertainty of the regulatory limit that we're currently using? So again, going back to this issue of biological organization, we developed a research program that went through looking at doing in silico structure activity analysis of all the compounds in the spill. There's about 10 different compounds. Looking at all the in vitro high-throughput screening data, bacterial mutagenicity. We studied it in C. elegans. We studied them in zebrafish. We then did some dermal sensitivity with several of the compounds and mixtures in mice because there was a concern about there being irritancy. We did repeat very short-term genomic screening, and actually we provided some high-quality prenatal developmental tox data using classic OECD GLP studies. So it really ran the gamut of data that we could apply to this, and we did this all in about 6 months. This shows you the range of different, there's a variety of different compounds. It was a tailored program that didn't look at everything in all situations. It was like a screening and linkage study. And basically, the summary findings were that most of them were actually pretty inactive, most of the compounds. MCHM had some activity at around 100 mg per kg per day, and this was quite high of where, relatively, a much higher level than where the advisory level had been placed. One of the key pieces of data from this was actually looking at the activation of biological pathways, very sensitive marker in the liver of the rats that we knew there were suggestions from the SAR that the liver should be a target. And it really showed that nothing was really happening until you got above 100 mg per kg per day. And so this really provided CDC with confidence that the decision that they were making on using that level to base their screening was really a pragmatic decision, very fundamentally based in some good quality science that was already existing in the literature, backed up by some additional data that was transparently provided to them to allow them to make that decision. So, this really focused on, like I say, on the MCH level of the 100 mg per kg. And it was really a focus on dose, as opposed to a focus on hazard. If we'd have had to go and evaluate every possible hazard that was potentially suggested, we would still be here working, we probably would still be working probably in 3 to 5 years. But because we focused on dose as the decision point, it allowed us to very quickly go in and focus on generating data that could help support or refute whether that was a good point of departure. So let me, in the last few minutes, just reflect on what I've learned over the last 20 years of being an NTP as it goes towards the EFSA's new strategy document and the visions they're going with. One thing I can say about the people of NTP is that they are mission-driven. That probably is the #1 thing I think that separates us from other parts of the federal government and academic scientists, in that it's all about the translation to public health. And if you can, and it's somewhat, you know, I read in the EFSA strategy, the culture of using impure science, what I call regulatory science, the applied nature of it, if that has to be ingrained in the culture to make sure that you're going in the right direction. Likewise, the team science approach, as opposed to the investigator approach. When you're an investigator getting your own money for your own research, that goes back to being a pure scientist. You're often driven by your curiosity. I'm a scientist and I'm curious, but I still want to make sure that my data is used. And that's a very different kind of mentality. And being in a team forces me that way. I have to think as a team. And if I'm going off in one direction, the team can bring me back centrally to, well, did you think about this? Maybe we'll go that way. The centralized funding of that really helps with that, because essentially we have capabilities that are shared resources. So immediately, if you're sharing resources, you have to come to a consensus that this is a good thing to be doing. I could come up with a great study, but because I'm sharing those resources with somebody else, they could say, that's a stupid idea. You should be doing this because this will address it. So again, it gets ingrained into the culture. Now, that said, when you have diverse drivers and a central funding, project management becomes very, very difficult, because you have many different stakeholders that you have to deal with. But the flip side is, by being headquartered within NIH, which is a non-regulatory organization, it's a biomedical research organization, it means actually that we actually still have the freedom to push boundaries of the kind of data that could be used for decision-making, rather than necessarily being bound by what we may normally be asking for. So you might normally ask for a certain type of study, and if you're within that particular regulatory authority, you may always just generate that kind of study. But by being outside of that, we can actually push the envelope a little bit. Finally, the thing that is probably ingrained within NTP is transparency and being open and being responsive to the public. All of the data that we generate on all of our studies is deposited within the SEBS database, the Chemical Effects on Biological Systems, and that goes all the way to the individual animal level data. When we do it on our NTP studies, for the apical studies, it goes all the way to the well data on the other in vitro data. What this means is other people can evaluate that data, and they do, and come up with different conclusions. That's okay. Different way of analyzing the data is perfectly okay. And similarly, when we generate our reports, they are all open and usually public peer review, because we don't have the monopoly on great ways to look at information. So we'd like to have public reviews. Literature analysis, we're starting to move into using systematic review rather than just expert panel review, simply because this is now creating objectivity in how you make those decisions. Again, transparency in the literature that's used, how different literature is weighted differently in the decision-making, and whether or not you agree with it, because part of a systematic review is based upon what is the PICO statement, what is the population, how are you going to go about doing it? So we put all that information out as well. Similarly, the kind of tools, we're developing new tools so that people can come to our website and actually do their own visualization of those data to make sure that, to see how they feel about it. And similarly, with ICVAM, how we're developing new data streams. We have public forums with people that, frankly, have not really liked the fact that NTP has used a lot of animals. We are working in dialogue with them to kind of come up with what is a pragmatic way forward to actually do things like validating those much more rigorously. So as you can see, transparency is a very important thing for us. And I'd like to leave with a quote from David Rawle, who was actually the director of NIHS at the time that NTP was created. And it is this: we basically exist to develop information and tools that both agencies of government and industry need so that we can all live together safely in the same world. And I think that seems, I heard echoes of that from the EFSA opening discussion, and this is what we do, and this is what we should be doing. And I'd like to thank you for your attention, and if you've got any questions, I'm happy to entertain them. Thank you.
So thank you very much. Hands up. Anyone want to actually make a very specific question to this presentation? I don't see it. Okay, yes, thank you for waving.
Alberto Mantovani from the Italian National Health Institute. I'm a toxicologist and I also have a panel member. Well, as an impure scientist, I have a technical question to another impure scientist. In an increasing number of cases, the results of apical tests to be used, or better, to be usable in risk assessment, they need mechanistic understanding. In some cases, even unconventional endpoints like, I don't know, obesogenic potential may need to be explored. How is the NTP coping with this need in a cost-effective way, of course, and what information, in your opinion, can trigger this mechanistic follow-up? Thank you.
Oh, that's a really, really good question. I mean, how, you know, mechanisms versus apical endpoints is a classic question for us. And I would say in the early days, we left the mechanistic understanding to others. I mean, we have a certain skill set. Generating high-quality GLP data was what we generated, and used that to then trigger and communicate with our extramural partners, who then go and fund academic researchers to let the academic community explore the mechanisms when there's a hazard that's been identified. Now that we're moving forward in terms of the TOX21 and generating essentially mechanistic data through the course of our screening, that is now starting to get incorporated into our actual thinking when we actually are pulling together the protocols and the strategy to assess something. So where we can, we're starting to use it where we have it, but I would say it's not the primary focus, because we can only do so much as an organization, and the world of academics, that is where mechanistic data can really come into play. But I think, as I pointed out with the Clarity BPA example, structuring that for very specific mechanisms is helpful, and the way we do that internally is we have dialogue with the program managers who do the funding through NIHS for extramural partners. I hope that answers your question.
Thank you. Any other question from the audience? If not, I would like to have a question, actually. I don't see any hands, but then if I profit of sitting there and making a very specific question related to the governance and budget management. So at the end, you have an entry point of requested. Who make the final decision? Who says, you stop this, and then you start something else? That's one question. The other, how is this linked to budget management? You said that the NHS has a yearly budget, but I suppose your projects run beyond 1 year. So what is the budget management on that? Or do you revise every year what is the outcome and what should be done next?
Yeah, so how do we manage the budget relative to different priorities? So like I said earlier, consensus building and consensus decision making is the way we manage it. Imagine you have a single pot of money. So the money doesn't come in for a given project. You have a baseline amount. When a new nomination comes in, all the management team are the ones that look, and then they're all impure scientists. We kind of look at that and we go, is this a priority? Is that a priority? Based on some of the issues that I was highlighting, what's the need? What's the question? What's the prevalence of exposure? How will the data be used? We basically use our logic model to look at whether we should actually proceed or not. And in some cases, you can go slowly on something and you just put it down on a lower priority. The decision is in-house, but other people know what we're working on. And so we can take that information when we put out a concept that we're going to look at naturally occurring asbestos, for example. We will get feedback from the public, from other stakeholders, from industry, and we take that into consideration. As also in part of developing those protocols, we send those to all of our interagency points of contact. So we have individuals within other federal agencies, and we say, this is what we're going to do. Should we do it? Have you got any other information that we might need? And so that allows us to make our own internal decisions, because we basically have felt that making some of those decisions internally somewhat separates us from influence. We can actually design the best science that's needed to answer the question. I hope that addresses that. Did that get all the questions there?
Yeah.
Okay. All right.
Yeah. then we can go back later.
Okay.
So thank you very much.